All right, welcome everybody. We will get going. We barely got into chapter 3, but we will get through this today and Friday. The exam is set to open Friday, scheduled right after lecture ends, and it's open until Monday at 5 p.m. So, any questions or concerns about that stuff before I get going with this? Okay. Oh. Can I repeat it, please? So I'm assuming exams details. The exam will open this Friday at 10 a.m. and it closes Monday at 5 p.m. There will be an ample window of time when you get on there. You have to complete it. I believe probably 2 hours. It should not take you 2 hours. Open book and open note, not open neighbor. Good. And hopefully good. Okay. So Monday, we wrapped up lecture talking about Francisco Reedy and his experiment with some meat and maggots. All right. And I kind of ended and I was like, but what about the microbes? What about all of the nasty sliminess that we would see on these steaks as they sat for a little bit of time? And I also was saying spontaneous generation was something that took a super long time to truly get everybody on board and say, that just doesn't happen, right? So Needham, he was a naysayer. He was pretty sure that those microbes that maybe we saw on that rotting flesh, they arose spontaneously, right? That must be, right? We can't see them and then it just shows up, even in the corked one, right? It must be some sort of life force. Spallanzani, he shifts gears and starts looking at some broth and hopes to disprove Needham, right? And basically, there's a lot, you know, when you're doing science, you would think there needs to be a little of this, right? We shouldn't just see something and accept it immediately. We need some other data, right? We need more stuff to back it up. So it kind of makes sense that this took a bit of time, all right? And they needed to have other experiments, similar but set up maybe a little bit differently, that helps to really solidify this, to give us more proof, more data to back it up, all right? So instead of looking at solid stuff, meat, he starts thinking about liquid cultures. Lots of bacteria that we're going to present to you guys in the lab are in some sort of broth culture. And then you typically take it out of that and put it onto a petri dish, some sort of solid media to get it to grow, right? So there are numerous things that we think about, and I'm not going to spout off all sorts of details with media. We'll talk a little bit about some of that stuff later when we think about some things bacteria might really like, right? But there are all sorts of things that we might think about using. There's stuff in your kitchen you could think about as different types of broths. Chicken broth or beef broth, vegetable broth, right? All sorts of different broths that you might utilize for something, right? So we could take a meat infusion, we could take those steaks and we could cook them up and get all of these juices and bacteria. They like that stuff. There's a lot of nutrients in there, right? And in turn could probably grow super good in some of those types of solutions, some of that food stuff that you're giving them, all right? So that's a point that becomes important when we think about this, all right? And he could cook up some broth and put some, allow it to be exposed to the air and see some stuff changing. We start with broth in the lab that looks, there's a kind of a tint to it, but it's see-through, right? It's clear, if you will. And as you inoculate it, as you put something in there, it gets very cloudy, turbid, and potentially super turbid if you've got a lot of good growth in there, right? So he could show some of this stuff, and it seemed to kind of make sense that it is something else, right? Not just this life force, but there's still issues. There's still naysayers. And in steps our good friend Louis Pasteur. So he did lots of stuff, all right? More than just pasteurizing milk, all right? Lots of things. And we'll really give him credit for being the final one to drive that nail in the coffin of spontaneous generation and to disprove it, right? There's technically another player that I'm going to talk about after him in 1 second, all right? Don't have a slide for him, but I feel like we need to mention him, all right? So Louis Pasteur came up with some ideas, basically going on the shoulders of what Spallanzani had set out to show, all right? But with some of these naysayers, they're thinking, well, it's the air, right? That's really what's doing this stuff. And Pasteur is pretty sure it could be things in the air, but not the air itself, all right? So he designs this flask, makes this what we call a swan neck flask. You can order up this type of glassware now, but he made it. He was the one that took a flask, this round bottom flask, and heated it up so that the glass became pliable, and he was able to curve it, to bend it into this kind of swan neck or U-shaped flask, kind of like the trap under the bottom of your sink to keep the sewer gas out, right? That's really what that trap is helping to do. Otherwise, it'd be a pretty stinky bathroom, right? All of the time, not just some of the time, right? So the idea with this is to provide an opportunity for his liquid to be exposed, to have the lid off, if you will, but to prevent things that might be in the air from actually getting into it, right? So he bends this flask, he's got his liquid in there, a meat infusion, all right? So beef broth or something like that is what we're starting with, all right? And he cooks it up, he boils it. And when you boil something like this, well, you pretty much are sterilizing it. There's nothing that should be growing in there. Pretty much nothing, all right? We'll get to that in a second, all right? So nothing should be growing in there, right? He's cooked the crap out of it. And now you've got this clearish looking broth exposed to the air, but things in dust, maybe microbes, can't get in there. They settle down at the bottom of our little U-shaped neck, all right? He takes another flask. He's got a handful of these flasks, all right? Takes another flask and does the same thing and heats it up, boils it, sterilizes it, if you will, and then he breaks the neck of that off, leaving it opened to the air, all right? And as it's able to sit for a few days, things in that air settle down into it, and in turn, it turns turbid, as we see this flask right here. But these ones stayed clear, right? He took some of them and he tilted them and let the liquid go down into the neck, touching that dust, bringing them back upright, leaving it all intact. And those things turned turbid as well, because they came in contact with the stuff that had settled down. Seemed like the perfect answer, right? It makes sense. We can see this stuff. There are microbes, there are small, invisible things floating around in the air. And given the chance, they could settle on our whatever it is and start to grow. And if it's full of good nutrients, they might grow really fast, right? In the lab, when you were doing your first stuff, thinking some environmental stuff. I don't know exactly how they have all of this set up, but there are numerous things that you could do. Or you could take a Petri dish, take the lid off of it, and just let it sit there on the bench for part of the time during the lab, or maybe put it up away. So it's not just here with me spewing out stuff, right? Away, so that we're not breathing right onto it. And then cap it up, allow it to incubate, and you will see there are all sorts of things just floating around from the air that are able to just land on it and start to grow. All right. So that's kind of what we're seeing. All right. And it seems to make sense. All right. And we say we give him credit, but deep down, there's still a little bit of pushback. All right. Because Pasteur's working with some meat stuff. All right. And I said pretty much everything's going to be killed by that. But there's some bacteria that cooking them up like that's not sufficient, right? And in turn, that's why we think about autoclaves or pressure cookers, heating things well above boiling point to kill other things, all right? So someone's like, yeah, it works with the meat, but when I made this vegetable broth or, some infusion, a hay infusion or something, I picked some stuff, cooked it up, allowed it to cool, and there's growth in there, right? What do you have to say about that, Louis Pasteur, right? I told you spontaneous generation happened, right? And in turn, what they really were looking at were some of those bacteria. I showed you the bacillus before, and the potential for them to form spores, right? Protective structures that allow those guys to live on another day when conditions become suitable. They were very insuitable when that broth was getting cooked up, and as it's getting hot, getting close to the boiling point, some of those cells technically are going to die, but a few of them were quick enough to replicate their chromosome, to package it up, and to basically protect themselves in these little spores. When that solution cooled back down, they were at a temperature that was good for them to exfoliate, to go back to the bacterial, the vegetative state, and start growing and feeding and replicating, and turned that liquid turbid, right? Still bacteria, still not spontaneous generation, right? But those naysayers, they were pretty sure, right? So there was another individual, and this is all pretty close to the same time frame. Tyndall, I want to say Joseph Tyndall, came up with a different process. And this technically is something that still could be used. Most of us wouldn't go through this process at our house to try to cook something up, all right? Trying to can some green beans with some Tyndallization, probably not something you really want to do, all right? So Tyndallization from Tyndall is a process of cooking something like a vegetable broth to sterilize it. And this is something that was done before we have a pressure cooker, before we've got the ability to heat things up above the boiling point, all right? So the bacillus, the bacteria that could be forming spores and contaminating stuff, like I said, when you first start to cook it, some of those cells are gonna die, right? They're not quick enough to form those spores, but a good chunk of them do. So I heat it up, I kill a few of them, some of them form spores, and I cool it back down. As I cool it down, those that form spores go back to forming the vegetative bacteria, the active, and I heat it back up. And as I do that for about 5 days, by the time I'm done with that, any of those bacillus that were in there, I've ultimately cooked them all. I've killed them all, right? But it took a super long time, right? But he was able, with this process of heating over the course of about a week, to create a vegetable broth that truly was sterile. And then it mirrored exactly what Pasteur had set up with the beef broth, all right? And he did the same experiment and opened one up, left one closed, tilted it, did all of the same stuff and showed. This is really where it's coming from. It was a little bit later. The endospores were actually identified and visualized, and we recognized it's these crazy bacillus that are all over, right? They are environmental bacteria. You can see all sorts of produce that has that stuff on them. And typically, it's not a big deal at all, right? Those that potentially may cause issues, usually you think about cooking that stuff and all fine and good, right? Other ones that might be on there aren't really going to cause any issues in the first place, right? And the cooking, we mentioned before, Bacillus cereus as a potential issue. There are some problems with it, and we'll talk a little bit about those guys later, but they potentially might be on rice. And you might cook rice, heat it up, and if it's not kept at the proper temperatures, not kept warm enough, or refrigerated promptly after you are done eating it, you give the opportunity for that bacteria that might be a spore in there to germinate and to start to grow. And that's how sometimes rice dishes can cause foodborne illnesses if they aren't kept at the proper temperatures, right? Just depends, right? And it all really goes back to that. So we showed Robert Hooke's stuff when we were talking about microscopy in the previous chapter, right? These images of the cork that he was able to observe and coining the term cells. That's really, as we're moving through here, what we want to think about, differences between cells in the first place, right? So we've got a couple individuals as we move in thinking the cell theory, all right? We have just disproven our ideas of spontaneous generation. We know that there are cells. We see images of these cells. Hooke's got all of these images of the plant cells, the cork, right? So Virchow and Ramak both together come up with these ideas that play on one another, all right? So the cell theory, right? That everything, every living thing is composed of at least a single cell, right? When we think multicellular organisms, potential for trillions of cells, right? But at least a single cell, right? And adding to it, and really, you know, driving that spontaneous generation doesn't happen. Life comes from life, right? Cells come from cells, right? Pre-existing cells, egg and sperm, unite to form us, right? Life from life, right? And everything that is alive is composed of at least a single cell encased in a plasma membrane, maybe some extra stuff, all right? So these two set the stage for these ideas. When we think more about it, we've looked a little bit, and we're going to go through more details with all of these cells. We've looked at prokaryotic cells, really kind of bare bones, and we want to think a little bit more about eukaryotic cells and sort of how is it that some of this comes to be in these cells in the first place. And I think a lot of this is really pretty fascinating. Typically cells are very small, all right? Microscopic for the most part, all right? There are some that are larger if we think about eggs, chicken eggs, or ostrich eggs that are humongous, right? Those technically cells as well. But when we think about in our body, very small, all right? And truly, as far as the size of these cells, they're microscopic because a lot of what's happening inside of these cells, they can't be gigantic and still maintain the functions that they truly need. Lots of nutrients move into these cells through simple diffusion, maybe some active transport. But once it gets into that cell, it's going to start moving around really, based off of concentration gradients spreading throughout that cell, right? The cytoplasm of our cells, lots of water, but there's lots of nutrients, lots of sugar molecules that are within there as well that are going to be broken down, right? And if your cell is humongous, that becomes super hard to get substances, whether it be nutrients or other molecules that we need for the generation of stuff inside of these cells. It's very hard to get those things to interact. But if you're constrained to a super small size, that becomes very, very simple, right? Very easy for that to happen. So, truly there's some physics behind this, surface to area volume inside of these cells can't be gigantic, can't be enormous because of some of those issues of really nutrient acquisition and dealing with all of this inside of the cell. They need to be little, all right? Bacteria shaped or size, or we think about eukaryotic size, about 10 times the size of a bacterium, but nothing that's much larger than that when we think about this type of an idea, all right? With the exception of, the chicken and ostrich eggs and so on, all right? That's a little different. So bacteria and eukaryotic cells are doing similar things, pulling in nutrients and doing all of these things, all right? But the evolution of eukaryotic cells, I like to think about this a little bit like, from a human perspective, not thinking like I'm a bacteria, all right? I'm just a machine for my bacteria, all right? But when I think about it from a human perspective, all right? Imagine it's just you, right? And then maybe, you've got yourself a little apartment, a little studio maybe. Then maybe you find yourself a sweetheart and move in together, right? Now there's two of you in this little place, right? Maybe that's okay. Maybe that's cute, right? Pretty soon you're like, you know what? We need a little more space, right? Just a little bit more, right? We can up and move, right? We can do all of that on our own, right? And just find something larger, right? So let's say we've got a little house and it's just the two, right? And then you decide maybe you want to have an addition, right? You want a little one around, right? So you go get yourself one, right? Now you've got one, right? What are you going to do about it, right? You've just got this little house, right? You could move, or maybe you could add on, right? You build an addition, right? You go out, right? We could do that with where we might live, right? But in a cell, we're constrained by that size. We can't get just continuously larger just because we've got more stuff going on, right? Now I got 5 kids, right? I need more space, right? I need many more rooms, right? We can't do that as a single cell, right? So instead of growing out, they grow in, right? They push plasma membrane to the inside, invaginations, moving the surface area to the inside, making more with the same size you got, right? Getting different little containers to put all of these things, right? As we form organelles inside of the cell, all right? As we start partitioning, right? They get a bit bigger, but they get a lot bigger by pushing these membranes to the inside, dramatically increasing their potentials to do stuff, right? To do more with less, all right? Or with the same, all right? So there's a lot of stuff happening, all right? And where does it truly happen with these steps as we're moving through? That's all really speculation, right? There are a proto-eukaryote here, which may have potentially already started this process of invaginations and making endomembrane system, all sorts of inner infoldings. And then we start thinking about these feedings, right? Or maybe we do it a little bit different, and we're pulling some of the aerobic bacteria in a little earlier. It's hard to say exactly how much of this stuff we've got going on when the other steps start happening, but it's likely this process has already started. All right. Life feeds on life. All right. For the most part. All right. Life feeds on life, and there's lots of good stuff there. All right. And bacteria are pretty awesome recyclers. We're going to touch into some of that a little bit later. All right. But this proto-eukaryote, primitive type of eukaryotic cell, there's all sorts of things that we think about more invaginations and issues, right? If we're doing all sorts of chemical reactions inside of the cell and potentially finding more nutrients and getting better sorts of energy sources and maybe breaking down things that potentially could maybe be issues, toxic to some of these substances, protecting your chromosome might be one of the first things on your radar. We want to keep these instructions pristine, right? And I don't want a bunch of acid products or something that is a byproduct of my consumption to destroy my instruction manual. That's bad news. So walling it off in a membrane helps to ensure that those instructions stay protected, right? And we start this formation of inner membrane, endomembrane systems, right? And endoplasmic reticula and all sorts of other things start to emerge, protecting and partitioning jobs. We're feeding still, and there are, the early organisms, microbes, were very different than we think about like life today, although there are still some of these microbes that function very similarly as these ancestral forms. Way back, as we think about the first types of organisms, they were anaerobic. Oxygen was very minimal. And in turn, they were able to exist and persist with other mechanisms, right? They were able to survive, right? And in an anaerobic situation, oftentimes you don't grow super quick, right? You're barely pulling in what you truly need, right? And some of these bacteria, we see some shifts. And we see our little green guys over here, our photosynthetic bacteria. Some of these organisms adapt and evolve strategies to utilize energy from the sun and carbon dioxide to build their own sugars. Now in this turn, they're feeding themselves, right? Not life eating life, right? This is feeding itself, right? And in the process of this, they start spewing out their waste product, which is oxygen gas. They spew out lots of it. And with the evolution of these photosynthetic bacteria, we describe it as the dawn of the oxygen revolution. This is what set the stage for what we know today, right? Were these very tiny bacteria, right, that start photosynthesizing. And as this happens, oxygen, it's technically toxic. And these anaerobic bacteria, they don't like that stuff, all right? And they, in turn, we may find some anaerobic organisms deeper in the crust of the soil, away from the surface where some of this oxygen could be killing them, right? They still have opportunities to survive, but others adapt strategies to deal with the oxygen, either making it so that it's not toxic or saying, wait a minute, I could use you, right? And in an aerobic situation, oxygen is used as the final electron acceptor. As they're pulling in all of this nutrient and breaking it down, we can utilize that oxygen. And in the process of dumping electrons and hydrogens to it, we generate water molecules, right? Now we've got aerobic organisms, and these suckers grow really good, right? And they can produce a lot more energy than their anaerobic buddies, right? And in turn, somebody eats one of them, right? And instead, I assume a good chunk of them probably get broken down and consumed. but some of them still inside of their vesicle start producing more energy. And that cell ultimately is like, wait a minute, if it could think that, right? I should keep you along with me, right? Instead of breaking you down. And they stuck around. This is the endosymbiosis theory, where mitochondria and chloroplasts ultimately emerged inside of eukaryotic cells. They were once bacterial cells, aerobic and photosynthetic cells that ancestral eukaryotic cells engulfed and ultimately kept around. The primary endosymbiosis is the engulfment of the aerobic bacteria. And the secondary endosymbiotic event was the photosynthetic ones, right? Everything has mitochondria. Pretty much everything has mitochondria. There are a couple exceptions with some of that stuff, all right? But for our purposes right now, it's all got mitochondria, whether we're talking animals or plants, right? But we shift over and look at plants, and they also have chloroplasts. If it was the opposite and the photosynthetic bacteria was the first to be engulfed, I envision some sort of sci-fi thing where we would all be photosynthetic. We would be little green Martians, right? It makes sense. And they're green because they photosynthesize, right? They also you don't see mouths on them. They don't need them. You don't need to breathe. You're photosynthesizing. You're a closed system, right? That would be awesome, right? You don't need to eat. You don't need any of that. You just step outside for a little bit, right? You get all you need, right? Maybe, right? So that seems kind of far-fetched, but could there be something like that? Could we manipulate skin cells to harbor something like that? I think that's pretty crazy, but kind of cool, right? Pack back, I feel it, right? What the heck did he talk about, right? All right. Very interesting stuff, all right? And there's other features that truly show these organelles that we see in these eukaryotic cells once originated as bacteria. Their chromosomes, they have their own chromosomes, and they are circular, not linear like the chromosome in that eukaryotic cell that they live in. They replicate on their own. They synthesize virtually everything that they need. They technically, through a symbiotic event, it's a both partner benefit sort of an idea. They have quit producing some of the stuff because the eukaryotic cell basically is like, I'll do that for you. And why waste your time, right? So they are dependent upon one another. They replicate their chromosomes, and they're different looking chromosomes. They've got ribosomes that are bacterial sized instead of eukaryotic sized. They were once bacteria, right? And in this endosymbiosis, we can't live without them, and they can't live without us, right? Pretty cool, I think. All right. Microbes. So flashing forward a bit, Ignaz Semmelweis, he did all sorts of things. And truly, some of the best things he did were save women's lives. Back in the day, if you were a woman and had a bun in the oven, in the back of your mind, you're probably thinking, am I going to live through this? And if you're not thinking that, you should have been, right? You didn't have very good odds of going into labor and delivery and actually surviving, right? There are all sorts of stories talking about some of these things. And, you think about, going into labor and you get a bag packed so you can go to the hospital, you got all your crap together, right? Sometimes it happens at odd hours and you're not thinking right, and you may pack things that you truly don't need, right? That's beside the point, all right? I will I will tell you this real quick, and then we'll go back to Semmelweis. With our first child, we had done all sorts of things. I went to some childbirthing classes, and we've gone through all of this stuff. Think that we're pretty well prepared. And a couple of weeks before the due date, bam, water breaks, and it's like 2 a.m, right? We are sound asleep, right? Not anymore, right? And now I'm freaking out, right? Because this is early. We're not prepared. I don't know what's going on, right? So as my wife is frantically trying to figure out what's up and, trying to gather some stuff, I decide I need to get some stuff too, right? We're going to need some snacks, right? I'm going to need some snacks. She's probably going to need some snacks, right? We got to the hospital, and I had a little bag full of cheese sticks, a dozen cheese sticks that we never ate a single one of. They got thrown away. There they sat in the room, right? And I didn't realize it until the next day. I'm like, what in the hell? Why did I bring these cheese sticks, right? Sometimes you just don't know, right? All right, enough of that. So you think you're getting ready, you pack stuff that you might need, and also they would pack another thing. They would prepare their death clothes, which I'm just like, how crazy, how disturbing, right? You're preparing for this, and you're also preparing to die, right? Because you got to pack your stuff. Your husband's not going to do that. He doesn't know, right? You pack it all, right? What? So Semmelweis is a doctor at a hospital where there are 2 wings to this hospital, and it's a teaching hospital. Lots of hospitals are like that, right? And so there are medical students that are working on patients, and they're working on cadavers. They're doing all sorts of stuff, right? And there's another wing that is where the midwives are. And when you, as a female, when you went to the hospital to deliver, you could go to the midwife or to the doctor's side, all right, and deliver your baby. When Semmelweis comes into play, he recognizes, and this is in all sorts of hospitals, but he recognizes that women that delivered on the doctor's side had a very increased rate of mortality compared to those that delivered with the midwives. And he started to wonder what's up with that, right? That seems weird, right? The doctors, there's issues with this as well, all right? So he makes some observations, and he sees some things happening that kind of makes him wonder, could this be the problem, all right? So the students are working on a cadaver, and a woman comes in to deliver, and they turn from the cadaver and they deliver the baby, and they go back to their schooling, right? They go back to the cadaver, and that's exactly how it worked. And probably similar instruments that they were using on the cadaver could have been used on her right at the same time, right? Maybe wipe them off, but why, right? This thing over here is dead and there's no issues with it, and I've got to deliver this baby. And then she dies, and so does the baby, right? Lots and lots of issues, right? Child bed fever or puerperal sepsis were the result of these issues, bacterial contamination, right? And he recognizes that that's not happening in the other wing, and those midwives aren't working on dead bodies. And guess what? They also wash their hands before they start doing their business delivering that baby. And he thinks, geez, maybe we ought to do the same, right? So he wants to instruct all of these doctors and soon to be doctors in the importance of hand washing. And just like spontaneous generation, there's a bunch of issues with that, right? He gets a lot of pushback. these doctors, prominent males that are pretty sure they know what's best, right? Who are you to tell me to wash my hands? My hands are clean, right? And in turn, it took another long time to truly get people on board to really wash your hands, right? And I'm gonna not click on this video. I haven't checked all of this stuff to see if I could do that yet. But there is an interesting video that talks about Semmelweis and some of these details. I recommend you check it out, all right? So a couple more players with the history of these things. Lister, this is more medical stuff, all right? He recognizes other issues, all right? And comes up with some ideas with sterilization and cleansing stuff to ensure that wounds were cleaned properly and that instruments and maybe the floor of the hospital, maybe that gets disinfected as well, right? And Listerine bearing his name, right? He's not the one that truly gives you that, but you know, the medicine-y kind of Listerine, it makes you wonder, right? Was that what they were mopping up the floors with? It tastes like it should have been, right? Super strong, right? You know that's gonna kill some stuff. You can just tell, right? You know, right? And I say, thank you, Joseph Lister, right? All right. So Robert Koch, he comes up with some ideas to truly look at this. And I think, did I talk about Koch's postulates before? No? All right. So his ideas were to look at some sort of infectious agent and truly say, this is the culprit, right? So a type of bacteria, anthrax or something else. Anthrax was one that he studied, and you could potentially see a wound, a cutaneous form of it, and scrape a little of it and inoculate it onto something, maybe like some agar dish or a potato slice. Lots of bacteria was grown on little pieces of potatoes before we start thinking about the media like we use today. So he grows this stuff up and then sees the bacteria on there, takes some of that, puts it onto a lab animal, makes a wound and inoculates it in there, and sees a similar looking wound as was on the patient, takes a scraping from that, inoculates it onto another potato slice, grows it up, and it all looks the same, right? Same bacteria here as there, same sort of looking wounds, and in turn, this is what truly caused this disease, right? We call it Koch's postulates, a series of steps like I just described to go through and say, this organism caused this infection, and this is how it looks, and I was able to replicate it, all right? So this is where we start thinking about germ theory of disease, right? He and Louis Pasteur are both right in the mix with this as we are talking about these microbes and the formation of all sorts of infectious disease, right? So here, shifting over, we can see a couple of radiographs, x-rays. This is an individual that's got pneumonia with the infiltrate in here, the cloudiness, and then an individual over here with normal lungs that are nice and clear. So lots of fluid within there and suggestive of that. So we're not actually saying any details in here as far as what culprit this is. Not at this point. There are numerous potentials for pneumonia. So quick timeline with some of this stuff as we move through here. The ancient Greeks proposed this miasma theory, this kind of foul air, bad air, these things, just you're contracting stuff just from the abyss, right? Spontaneous, but it's this, you know, this air that's doing it. We progress along from the ancient Greeks, 1500s, we start thinking a little bit about this with Fracastro, thinking initially about some germ theory, contagion, contagiosis morbus, these things, but still potentially from the miasma, but things that are causing these illnesses that we start thinking maybe there's a little more to it, right? Hooke starts seeing cells as we flash forward about a century. Ley and Hooke a decade later, seeing other organisms with maybe a little more sophisticated types of microscopes. And now we jump forward a couple of centuries with Semmelweis and the importance of hand washing. John Snow, and there are other videos that go through some of the details with him. We think of all, lots of these people as, most all of these males, fathers of something, fathers of microbiology or germ theory. John Snow, father of epidemiology, thinking about studying patterns and trends in the progression of disease. Cholera is a nasty bacterial infection. If you truly want to crap yourself to death, cholera is what you should get. It's a bad one, all right? And in turn, in London, there were a couple of really pretty nasty outbreaks of cholera that he was able to trace the spread through the water supply. And there was other issues with that, lots of pushback, because there were still people saying, nope, it's this miasma, right? It's not the water, right? It's not microbes within the water. We don't believe you, right? And it smelled terrible, right? And that smell, that's what got you, right? Not so much. All right. Pasture right around here as well, as we start thinking about pasteurization process and dealing with fermentation. I think I mentioned before, really his initial stuff wasn't for milk. It was more for beer, to keep it from fouling. And then we're like, wait a minute, we could use this for other things, right? So, and he's listed repeatedly. So, and then Lister and Koch, as we left with them a second ago. All right, I think that, a couple minutes early, but I think that's a perfect stopping point. We will pick up with the last of this on Friday. Farewell, good day.